is double-stranded. There is an anti-parallel arrangement of the sugar phosphate backbones when from the 5' end, which is characterized by a phosphate, to the 3' end, which is characterized by a hydroxyl that is in the 3' position of the ribose of the DNA. This is important because its DNA is deoxyribose, because in the 2' position, as you recall, there's a hydrogen here and not a hydroxyl. What you will also remember is that there seems to be a direct correlation between the amount of thymidines that you find in DNA and the amount of adenines, as well as the number of guanines and the number of cytosines. And that's because adenine and thymine hydrogen bond together through 2 hydrogen bonds. Guanine and cytosine form 3 hydrogen bonds. And if you recall, this actually makes guanine, cytosine, hydrogen or base pairs a little bit stronger than the ATP base pairs. Okay. But I also want to point out to you, and I'm sure you learned this when you were doing your molecular modeling lab, that DNA has a major groove and a minor groove. A major groove and a minor groove. Okay. All right, so this is the overall structure. But early on, the actual structure of DNA wasn't fully realized until about 60 years ago, which isn't that long, you think, in terms of scientific ideas developing. It was known by the work of Chargaff that, as I indicated earlier, the amount of adenine in DNA seemed to equal the amount of thymine, the amount of cytosine, equal the amount of guanine. And also, back in the 1950s, there was a scientist, she was an X-ray crystallographer, Rosalind Franklin. Rosalind Franklin was working on establishing the structure of DNA. She was an X-ray crystallographer, and the structure that was deduced from her work is shown here. And what it shows actually is the double helix itself. That's the double helix itself. What was interesting is in Rosalind Franklin's lab, Jim Watson actually went to visit. And while Rosalind wasn't looking, Jim Watson got into her notebooks and found her structure. Because Jim Watson was familiar with X-ray diffraction patterns, which is what this is. This is an X-ray diffraction pattern from a crystallographic structure of nucleic acid. Jim Watson was used to looking at diffraction images, and immediately looking at this image, he knew that there was a double helix involved with the DNA. Then he quickly closed her notebooks and pretended he didn't know anything. Well, he got the Nobel Prize for that sneaky action. And everybody knows it too, but you don't take Nobel Prizes away. But Rosalind should have got something because it was her work that actually established the foundation. Unfortunately, at the time, X-ray crystallography experiments were very dangerous. There were a lot of X-rays that damaged the tissues of researchers working in this field, and she expired of cancer as a result of her work. Being exposed to X-rays, she died of cancer, I think, back in the 1950s, before she could be properly honored with the prize, along with not only Jim Watson, but Francis Crick as well. So Watson and Crick got the Nobel Prize, but it was really Rosalind Franklin. Her work actually laid the foundation for what Jim Watson was able to deduce. And that is, from this, he knew that DNA was a double helix. And prior to establishing this, it was known, again, that the amount of adenine equaled the amount of thymine, the amount of cytosine equaled the amount of guanine. But what they didn't know was whether or not you had purine to purine that would be binding together to make a double helix. This this wasn't understood. So it actually could have been guanine and adenine together, or pyrimidine to pyrimidine, that would be thymine to cytosine. And so when Jim Watson saw this and started doing the measurements, he knew that purine to purine was just too wide for the measurements that were coming out in Rosalind Franklin's X-ray diffraction pattern. He knew that pyrimidine to pyrimidine would have been too narrow for the base pairing that would hold the two sugar phosphate backbones together, or the two strands together. But like Goldilocks and the Three Bears, purine to pyrimidine was just right. It was absolutely consistent with the X-ray data. And so that was how the structure, the final structure of DNA was established by hook and by crook. Okay. And it made sense. That was one of the most important things about establishing the structure of DNA, is it made sense that the way that the base pairs came together, and also the structure of the molecule itself, that this would be the genetic material. And also prior to really establishing the structure of DNA, there was still some controversy over whether or not it was protein that was actually the genetic material and not DNA. Okay. All right, so now we're going to talk a little bit about what the results of establishing the structure of DNA did for then determining how DNA was actually replicated. Based on the work by Watson and Crick and Rosalind Franklin, it was known then that DNA was a double-stranded helix with guanine base paired to cytosine, adenine to thymine. And therefore, it was able to be determined that in DNA replication, these two strands would be separated. And then you would have DNA polymerase that would come in and put a base across from each base in the template strand. So these parental strands, the parental DNA molecule, would be considered DNA templates, and the polymerase would put the appropriate base across from each base in the template strand to make two new DNA molecules. This is called the semi-conservative. This is the semi-conservative model for DNA replication. Because it's semi-conservative because one of the new strands is newly synthesized, and the other strand is the template strand or the parental strand. Can I repeat anything or explain anything again? Are we okay? What? I'm sorry. Yeah, so the question is, this is semi-conservative, and yes, this is semi-conservative model. This is the semi-conservative model for DNA replication, and again, because one strand is newly synthesized and the other strand is the parental or template strand. And if you recall from our lecture on nucleic acid synthesis, that in the process of generating a new strand of DNA, what happens is DNA polymerase will bind to a nucleotide base, and the nucleotide base, of course, has three phosphates, alpha, beta, and gamma. And in the process of transferring the base into the growing DNA strand, the beta and gamma phosphates are hydrolyzed away, and it's the energy associated with this hydrolysis and the repulsive force between the phosphates that actually gives DNA polymerase the energy to help establish this covalent bond between the 3' hydroxyl of the base that's already incorporated into the nucleotide strand, and adding the new base at its phosphate group to that 3' hydroxyl to generate, if you recall, a phosphodiester linkage. So it's a phosphodiester linkage that's generated between the alpha phosphate and the 3' hydroxyl, the base that's present in the growing nucleic acid strand. And is this a dehydration synthesis? No, it is not. It is a phosphoryl transfer. And remember, your book is not right in that. This is a phosphoryl transfer. It's a phosphoryl transfer. It is not a dehydration synthesis. Water molecule is not split off in this process. So it is a phosphoryl transfer that is energized by the repulsive forces between the phosphate groups that are being split off as that new base is incorporated in, and that energy is used by DNA polymerase to actually seal that covalent bond between the phosphate and the 3' hydroxyl to form a phosphodiester linkage. Okay? And what I also want to stress is that DNA is generated, as well as RNA, the DNA is generated 5' to 3'. It's generated 5' to 3', and the template strand, of course, is anti-parallel to that 5' to 3' synthesis by DNA polymerase. So the template strand is going to be in the 3' orientation. The newly synthesized DNA strand, of course, is going to be 5' to 3', anti-parallel to this template strand. I'm just pointing out again what we went over a long time ago when we talked about DNA structure. Does anybody have a question, or can I repeat anything or explain anything further? We okay? All right. And in our last lecture, when I was talking about what happens during S phase, we had a brief look at synthesis of DNA in bacteria, where I talked about origins of replication. Well, in our own chromosomes, in the chromosomes of eukaryotes, origins of replication are also used for DNA synthesis. And this is actually a transmission electron micrograph showing origins of replication along the chromosome of a Chinese hamster cell, which basically uses the same mechanism as our own cells for replicating DNA. It's a little bit easier to actually look at the process with the diagram as opposed to the electron micrograph. And what you see here is during S phase, what will happen is there will be what are called replication bubbles that will open up along the length of our chromosomes. So replication bubbles will open up along the length of our chromosomes, and these replication bubbles actually open up in regions that are called origins of replication. Origins of replication are segments of the chromosome that are actually attached to parts of the nuclear matrix. Remember, our chromosomes aren't free-floating in our nuclei. They're actually attached to two components of the nuclear matrix. And what establishes an origin of replication is an attachment to the nuclear matrix. So during S phase, what will happen is the DNA helix is going to be opened up to form two single-stranded regions. And then what's going to happen, that single-stranded DNA, each single strand, is going to then become a template for DNA polymerase. And the polymerase will go in both directions from the replication bubble. And eventually the replication bubbles that have formed all along the length of the chromosome are going to blend together to form two new DNA molecules, one with a template strand, one with a newly synthesized strand. And again, this is the semi-conservative model of DNA replication. E. coli also use what are called most of what we know about DNA synthesis is based on studies of DNA polymerase and the components of the replication process are based on, that we know, are based on studies in E. coli. These are all based on studies in E. coli. Studying DNA replication in mammalian cells is much more complex because we have more variations on DNA polymerase. A lot of the DNA polymerase, and this isn't in your book, but a lot of the DNA polymerases that are present in the nuclei of our cells are actually involved in repairing DNA. And so consequently, we have at least 12 known different forms of DNA polymerase that are functioning within our cells, in contrast to a more simplified model that you find in E. coli, where you have DNA polymerase 1, 2, and 3. Okay? All right. So I just wanted to point out that in E. coli, you have a very similar situation where you have an origin of replication, and that origin of replication is actually attached to the plasma membrane of the bacterial cell. There's no nucleus, but it's actually attached to the plasma membrane of the bacterial cell. And then, of course, there's a replication bubble that forms, and then DNA polymerase will come in and start generating new strands of DNA off of the parental template strands. And eventually, as the polymerase works its way all the way around the molecule, eventually you'll have two daughter molecules of DNA, and bacterial chromosomes are round. They're circular. In contrast to our own chromosomes that are linear, our chromosomes are linear. Bacterial chromosomes are actually circular. And like I said, they attach to the plasma membrane of the bacterial cell. But take a look. Very similar structure to what we have with our own cells, or mammalian cells. Okay. All right, now I'm going to go into what has been determined about what is used during DNA replication, and a list of the proteins that are used in bacterial DNA replication. So like I said, this is where most of what we know comes from, studies on E. coli. And it's known that the first thing that an E. coli cell has to have to replicate its genome, or its DNA, is a helicase. A helicase actually helps to unwind that double helix. So the bacterial double helix that makes up its DNA, that DNA double helix, has to be unwound in order for DNA polymerase to bind to the template strands and generate a new strand. So it's the helicase protein that's involved in this process. And we have this too. We also have helicase in our own cells. Then there's a single-stranded DNA binding protein. So when the helicase actually opens up, the DNA at that origin of replication, then a single-stranded DNA binding protein is going to hold on to that DNA and keep it from clamping right back down to form a double helix again. So that's what the single-stranded DNA binding protein is for. It's to keep that helix open. And you can see the diagram of the single-stranded DNA binding protein. It's right in this position here. The helicase goes forward. We also have, not only in bacterial cells, but our own cells, an enzyme called topoisomerase. So when the helicase is actually unwinding the double helix, there's a lot of what you would call strain, winding strain in the chromosome itself. And so what topoisomerase does is it actually breaks the covalent bond along the sugar phosphate backbone, and it relieves that strain in the unwinding that has to occur. And we actually have two different types of topoisomerase. Topoisomerase 1 will actually break one strand. Topoisomerase 2 will break both strands of the DNA if the unwinding strain becomes too great. And topoisomerase also has the ability not only to cleave that covalent bond, but it can also rejoin it as well. Very important. Okay. And when our cells are in S phase, it's been found that there are at least 10 double-stranded breaks that happen across our genome during S phase. These double-stranded breaks have to be carefully repaired during S phase, or there's a real problem. Okay, and I can go into that later on, but I talk about cancer. Then we have primates. It's interesting that DNA polymerase can't just bind to DNA and start incorporating nucleotides. DNA polymerase actually has to bind to an RNA primer, and interestingly enough, it recognizes an RNA primer that is formed on the DNA template. And that RNA primer, of course, is generated by an RNA primase. It generates an RNA primer in a 5' to 3' direction. And once that RNA primer is put into place, then DNA polymerase 3 actually comes in and will start generating a new DNA strand. DNA polymerase 3 is a very large complex. It has what you call a clamp. So it forms what is called a sliding clamp structure on the DNA, on the parental strand, and incorporates nucleotides, like I said, in a 5' to 3' direction. Then what will happen is the RNA primer that started DNA polymerase along its way, that RNA primer cannot be allowed to remain. And so DNA polymerase 1, what it does is it removes these ribonucleotides, and replaces them with deoxyribonucleotides, or DNA. And then we have another protein called DNA ligase, and what that does is it establishes that final covalent bond when these RNA nucleotides are removed and DNA is put in, then ligase actually seals the sugar phosphate backbone. So these are the primary proteins that are involved in DNA synthesis. And what I'm going to do is I'm going to go over the two components of DNA synthesis. DNA synthesis happens through what is called leading strand synthesis and lagging strand synthesis. So when that origin of replication opens up and that replication bubble forms, what you want to see is the RNA primase will put a primer in, and then DNA polymerase 3 will bind to that RNA primer and then go in a 5' to 3' direction into the bubble, into the what is called the replication form, into the replication form. That's leading strand synthesis. It happens as one long strand. However, it's the opposite strand that can pose a problem, because as that replication fork is opening up, yes, the DNA polymerase can move in as the replication fork unwinds. But the problem is, right at the replication fork, where you have to have the other strand synthesized, this is not a very big space. It's not a very big space, and so consequently, this is called lagging strand synthesis. It's not continuous like leading strand synthesis. The other strand or lagging strand of synthesis occurs in 200 base pair fragments. Happens in 200 base pair fragments, and those are called Okazaki fragments. They're called Okazaki fragments, and that's mentioned right here. Okazaki fragments because it was a Japanese scientist by the name of Okazaki who worked out this model of DNA synthesis for the lagging strand and got the Nobel Prize for that. Isn't that nice? All by himself, as far as I know. Okay. All right. So in the case of weaving strand synthesis, which is the simpler process, you have an RNA primer, then DNA polymerase will come in and start synthesizing DNA in a 5' to 3' direction at the replication fork, and that's what this is. Single-stranded DNA binding protein keeps it open, helicase opens it, and further on up, it's not shown here, topoisomerase is actually cleaving the phosphodiester backbone, the sugar phosphate backbone, I should say, and